Hi everyone, I am Assistant Professor Mr. Manohar Kengar, Nootan College of Pharmacy, Kavathemahankal. Today's we are the learn Semisolid Dosage form, ointment, creams, and to the gel. Introduction: Pharmaceutical semisolid dosage. The preparation include ointment, paste, cream, plasters, gels, and the rigid forms. They contains one or more active ingredients dissolved or uniformly dispersed in the suitable base and any suitable excipient such as emulsifier, viscosity, increasing the agents, antimicrobial agent, antioxidant, or the stabilizing agents, etc. The semi-solid dosage forms are dermatologist preparations intended to the applied externally onto the skin to the produce local and to the systemic effect. The other dermatological products include the solids such as dusting powders, liquid dosage forms like liniments, and to the lotion. Definition: the semisolids are the topical dosage form used for the therapeutic, protective, or the cosmetic function. They may be applied to the skin or used nasally, vaginally, or the rectally to the produce local or the systemic effect. The advantages of the semisolid dosage form: first, it is used externally. Probability of the side effect can be reduced. First, fast gut and the hepatic metabolism is avoided. Local action and the site of the specific action of the drug on the affected area. Convenient or the unconscious patient or the patients having the difficulty on the oral administration. Suitable dosage forms are the bitter drugs, more stable than the liquid dosage form. This is the advantages of the best advantages of the semi-solid dosage form to comparison to the other dosage form. Because of it can be reduces the side effect and avoided to the hepatic and the metabolisms. It's avoided. Local action, the site specific action of the drugs at the affected area. Those are the unconscious patients and the difficulty to the consume the tablets and the other liquid dosage forms. Those patients are the used as the semi-solids dosage forms, and it is more stable than the liquid dosage form. The disadvantage of the semi-solid dosage form: first, there is a no dosage accuracy in the type of the dosage form. The base which is used, the semi-solid dosage forms can be easily oxidized, may causes the staining. They are the bulky to the handle. The application with the finger may be causing to the contamination. Physicochemical less stable than the solid dosage form, maybe causes the irritation or the allergy to the some patient. This is the disadvantage of the semi-solid dosage form. No dosage accuracy means it is not be unidosage form. It is easily oxidized, means the semi-solid dosage form the contain water content, and it is the chances to the occurring to the free radicals, which is the difficulty to for the skin absorption, and which is easily oxidized. Second one is the staining. It is a bulky to the handle. The application with the finger may be causes to the contamination is the major disadvantage of the semi-solid dosage form. This is the physicochemically less stable than the solid dosage form, and it is the irritation and to the allergy. Some of the patients were using the repeated to the semi-solid dosage form to the apply to the skin, then occurring to the allergies. Some of the patient. This is the main advantages of the semi-solid dosage form. The ideal properties of the semi-solids. The physical first is physical properties, which is the smooth texture, elegant appearance, no dehydrating, non-gritty, non-greasy, and non-staining, and non-hydroscopic. This is the ideal properties of the semi-solid dosage form. Second one is the physiological properties. First, take as non-irritating, do not alter membrane skin functioning, miscible with the skin secretion, have low sensitizing agents. The semi-solid preparation making to the it is a care to the some of the ingredients or the some excipients and the drugs which is the not irritating, do not alter membrane to the skin functioning, and miscible with the skin secretion, and the low sensitizing index which is a properly using which is the physiological properties. Second one is the application of the properties of the semi-solid dosage form, which is the easily applicable with the efficient drug release and high aqueous wash ability. This is the measures care for the preparation of the semi-solid dosage form. The preparation of the semi-solid dosage forms, which is the using the ingredients used in the preparation. First is the major using the ingredient of this the semi-solid dosage form. It is the base. Second one is the preservative, humectant, antioxidant, emulsifier, gelling agent, permeation enhancer, and the buffer. There is a 8 major ingredients are used in the semi-solid preparation. It is a base. First, it is one of the most important ingredient used in the formulations of the semi-solid dosage form. The ointment base do not merely act as the carrier of the medicament, but they also control the extent absorption of the medicament incorporated in them. The ideal properties of the bases, they should be inert, non-irritating and non-sensitizing. We have the choosing the base, which is the selecting base, which have the inert, should not irritant and non-sensitizing, compatible with the skin pH and to the drug, which is the base is the compatible with the skin pH and to the drug, which is the good solvents and the emulsification agents, which is the shows always shows for the solvent and the good emulsification agent, which shows the emollient, protective, non-greasy and easily removable, release the medicament readily at the site of the applications, and which have the pharmaceutical ingredient the process of the good stability. This is the ideal properties of the bases. The classification of the base, the ointment base are the classified by the USP into the 4 general groups. First is a hydrocarbon base, oleoginously base, for example petrolactam and paraffin lanolin bases, which are the used. Second one is the absorption base, which are the using the base cold creams and night resolution. C, water removable bases, which are the using the oil in water base. D, water soluble base, which are the using the polyethylene glycol, which are the used in base with in the ointment base. The second ingredient is the antioxidant. The oxygen is a highly reactive atom that is capable of becoming the part of the potentially damaging the molecules. Commonly is called as free radical. Free radicals are the capable of the attacking the healthy cell under the body, causing them to lose their the structure and the function. To prevent this, the antioxidant are to the added. For example, is the butylated hydroxyl ionides and the butylated hydroxyl toluene. These are the which are the used in the ingredient into the semi-solid dosage form, like the antioxidant. The formation enhancer, skin can act as the barrier with the introduction to the various penetration enhancer. Penetration of the drugs through the skin can be improved. Sometimes you have the using when the apply into the skin, it is the not be the cross to the barrier and not to the local effect. Then we have the using the formation enhancer, and then it is the help to the cross to the barrier and fastly local effect to the systemic circulation, which is the best part and ingredient to the formation enhancer into the using to the semi-solid dosage form. The second one is the emulsifier, as the emulsifier is a substance that stabilizes the emulsion by the increasing its the kinetic stability, must reduce the surface tension for the proper emulsification, prevent the coalescence, the ability to the increase the viscosity at the lowest concentration. Second is the emulsifying agent, sodium urlyl sulfate O/W emulsion, sodium citrate and the calcium citrate, which are the used in the emulsification agent in semi-solid dosage form. Glyceryl monocitrate, weak W/O emulsifying agents, which are the used in the stabilizing agent and emollient in the O/W emulsification, which the shows of this the emulsification agent, stabilizing agents and the emollient the shows of the O/W emulsion, which are the using in a semi-solid dosage form. Second one is a humectant. The humectant is a hydroscopic substance. The humectant are the used to increase the solubility of the active ingredient. Sometimes the drugs are the not be fastly soluble into the bases. Then when you have the using the humectant, which is the flow and to the get the high increase of the solubility of the active ingredient. To evaluate it is the skin penetration as well as to evaluate the skin penetration, evaluate the hydration to the skin. The humectants work like the magnet, the draw into water from the environment as well as from to the deep the within to the skin. The second one is a vapor. The vapor are the added for the various purposes such as the compatibility with the skin, drug solubility, drug stability, improves the ionization of the drug, skin due to its the weak acidic nature, tolerate the weak acid the preparation. For example, sodium acetate, sodium citrate and the potassium, which are the used in the semi-solid dosage form. It is compatible to the skin, drug solubility, drug stability, which is the most important factor for this vapor solution. That's why the using for the semi-solid dosage form. Second one, antimicrobial preservative. The antimicrobial preservative using to the semi-solid dosage form, because of which is the inhibit the growth of the contamination of the microorganism. So required the addition of the chemical antimicrobial preservatives to the formulation. For example, is a para-hydroxybenzoate, phenol benzoic acid, sorbic acid, quaternary ammonium salts, and the other compounds which are the using to the semi-solid dosage form, and which is the avoided and the prevention to the growth of the unwanted microbial growth into the semi-solid preparation. The first is ointment. Ointment are the homogeneous, translucent, viscous semi-solid preparation. Most commonly are is a greasy, thick oil. Oil is 80% and water is 20%. Intended for the external application to the skin and the mucous membrane. They are used for the emollient, protective, therapeutic, and the prophylactic purpose, which are the used in the ointment. Second, the classification of the ointment. First taken the epidermic ointment, B is endodermic ointment, and C is a didermic ointment. First is the epidermic ointment. These ointment are the intended to the produces the actions on the surface of the skin and the produces the local effect. They are the not absorbed. The epidermic ointment which are the not absorbed into the skin, which is the as used as protective, antiseptic, and the parasites. Second one is the endodermic ointment. These ointments are the intended to the release of the medicament that the penetrate into the skin. They are the partially absorbed and the act of the emollient, stimulant, and the local irritation, which are the using the endodermic ointments are the penetrate to the skin, as well as it is shows the emollient, stimulating, and the local irritation. Second one is the didermic ointment. These ointments are the intended to release the medicament that passes through the skin and produces the systemic circulation. The second one is the anatomy of the skin, the structure of the skin. The skin is consists of the three layers. The outer layer is called as the epidermis, the middle layer is the dermis, and the innermost layer of the hypodermis, the subcutaneous tissue. Thus, the epidermis does not have any direct sources of the blood venous to provide the nutrients. The protective keratinized layers is waterproof and the airtight. It prevent more substances to the intake to the body or the loop from to the body. Stratum corneum is the outermost layer of the epidermis and has the thickness of the 10 to 20 micron when it is dry. At the dry state, it is made up of the 75 to 85% of the protein, 50 to 20% of the lipid, and 10 to 50% of the water. Thus, the structure of the stratum corneum provided to the barrier. Dermis is the main mass of the skin, consists of about the 18% of the protein. Blood vessels found in the dermis provided the nutrients for the both the dermis and the epidermis. Sweat glands, especially erkinite, the sweat glands, subcutaneous gland, and they're the hair follicles. The end for the epidermis, but they the stem from the dermis. The second one is the preparation of the ointment. Both of the large and the small scale, the ointment are prepared by the three general method. First is incorporation method. Second one is the fusion method. Third one is the emulsification method, as well as involving the chemical reaction. The methods for the particular preparation depending upon the primarily upon the nature of the ingredient. This is the formulation methods for the preparation of the ointment. We are the learning to the step by step. The first is incorporation. The components of the ointment are mixed together by the various means until the uniform the preparation has been attempted. On a small scale, the pharmacist maybe mix the components of the ointment in a mortar with the pestle and the spatula and the ointment slab may be used to the rubs into ingredient into the together. The incorporation method is easily method. We have the using the mortar pestles and the spatula and the ointment slab and the user to the rub to the ingredient together. This is the flow chart of the saturation method. First upon we will the size reduction of the product of the ingredients of the drugs. Second one is the ligation. Third one is the mixing with the bases. Fourth one we have the using the all ingredients and the excipients with the saturate and the saturation. Second one is the mix bases, the produces the final weight of the product. Second one is the homogenization, and second one is the filing. This the all of the procedures we are the getting to the preparation of the product of the semi-solid dosage form. The second method is the fusion method. By the fusion method, all or the sum of the components of and the ointment are the combined by the being the melted together and the cooled with the constant stirring until the coagulated. Those compounds not melted are the generally added to the congealing mixtures as it is being the cooled and to the stirrer. The naturally heat level the substances and the any volatile components are the added last when the temperature of the mixtures is slow enough not the causes the decomposition of the volatilization of the component. The all the sum of the components the ointment combined together with the melt together called as cooled and the constant stirring until the coagulated. The not melted generally added the congealing mixtures as the being to the cooled and to the stirrer. This is the temperature of the most slow enough not the causes the decomposition volatilization to the component. This is the flow chart of the fusion method. First upon the grate the waxes the selected bases and the put out into the water bath. Then it is a high melting point at the first wax at low temperature and the melt into the bases together. Dissolving the oil and the soluble drugs added the small volume of the phases, the large volume of the phases. Then we have the cooling the under the stirring, soft massage is the 40 degrees Celsius. Second method is a homogenization and the filing. The third method is the emulsification method. This method in the preparation of the ointment having an emulsion type of the formula. The general method of the manufacturing involve the melting process as well as the emulsification process. The second one is the evaluation of the topical dosage form, the evaluation of the ointment. First up one is penetration evaluation, rate of the release of the medicaments, absorption of the medicaments into the bloodstream, and the irritant effect. This is the evaluations of the ointment in the topical dosage form. First upon the penetration, this is the simple method. The weighted quantities of the ointment are the rubbed over the definite area of the skin for the given length of the time. Thereafter, the unabsorbed ointment is collected from the skin and the weighted. The difference between the two weighted roughly represented to the amount of the absorption. This is the simple method of the penetration. We have the using applied into the skin of the ointment, and then the after time we have the collected the unabsorbed the ointment and the we have the using the weighted and to the get to the result the how much the penetration to the drugs into the skin absorption. The second method is the rate of the release of the medicament. The access to the rate of the release medicament, small amount of the ointment can be placed onto the surface nutrient to the agar media contaminated into the petri dish. Next one, if you have the medicament is a bactericidal to the agar plates, is the previously seeded with the suitable organism like the oasis. After the suitable period of the incubation, the zone of the inhibition of the measured, the correlated with the rate of the releases. When we are using the petri dish and out of the put out the oasis, the bactericidals and the using the ointment, then the how to the correlated with the rate of the release and the we have the getting to the result. The second one is the rate of the release of the medicament. The simmer the internal surface of the test tube with the thin layer of the ointment. Fill the tube with the saline or the serum, and after the gap of the time estimating the amount of the drug present into the serum and to the saline, we are getting the evaluation of the ointment. The last one is the absorption of the medicament into the bloodstream, which is a definite amount of the ointment should be the rubbed through the skin under the standard condition, and the medicaments are the estimate into the blood to the plasma urine. When you have the using the applied into the skin the ointment in the specific area, and then we have the collect the estimate the blood plasma and the urine and the tested the how amount of the drugs are the released into the urine and to the blood plasma. The second one is the last one, the irritant effect. The irritant effect can be also be judged to the certain extent by the injecting the ointment into the tight muscles and the under the abdominal skin of the rats. The reaction are the noted to the intervals of the 24, 48, 78, 72 and the 96 hours. The presence of the patches onto the skin within the 2 weeks they indicate the irritancy, the presence of the skin. These are the irritating the effect the help to the using to the rat. The second preparation of the creams. The creams of the homogeneously semi-solid preparation consisting of the opaque emulsions contain the lipophilic emulsifying agents. There are the consistency depending on the type of the emulsion, either the water in oil and oil in water, and on the nature of the solid into the internal phase. The cream are the intended for the application to the skin or the certain mucous membrane, which are the using for the specific for the protective, therapeutic and the prophylactic purpose. Second one, the classification of the cream. First upon the cream containing the microspheres, the vitamin A cream 200 and 215 microns, the laminar phased cream, the liquid paraffin and the water emulsion, the cream containing the liquid nanoparticles, the W/O cream and the more coals. This is the classification of the cream. Second one, the preparation of the cream. There are the 4 steps of the preparation. The preparation of the oil phase, the flax and the powder. The ingredients are dispersed in the mineral oil and the silicone oil. The heating may be required for the melting. Second one is the hydration of the aqueous phase, the emulsifiers, the stabilizer, thickeners and the dispersed in the water heating may be required for the hydrating. Third one is the forming the emulsion of the 2 phases are the blended under the vigorous agitation. The last one is the dispersion of the active ingredient. These are the 4 steps of the preparation of this cream. The evaluation of the cream. The first upon is the rheology. The rheology or the viscosity should be remain the constant. The rheologic measurements are the utilized to the characterized easy to pouring from the bottle, squeezing from to the tube or the container, maintaining the product shape in a jar, or the after extrusion the rubbing the product onto the skin. The viscosity can be measured using the viscometer used for the such of the liquid. This is the evaluation of the cream for the rheology. The second one is sensitivity, as the various type of the ingredient are used in the occasionally used for the antiseptic and the hormones, etc. There is a possibility to the sensitization or the photosensitization of the skin. This should be the tested beforehand. This test is normally done by the patches test on the skin and can be either open or the occlusive. The test sample applied along with the standard market product at the different places and the effective compared to the after the period of the time. C is the effect thermal stress. It is the usually evaluate the stability and the emulsion by the subjecting it to high and the low temperature in an alternating cycle. The sample are the first exposed to the 60 degrees Celsius for the few hours and the 0 to 40 degrees Celsius. Such exposure are the repeated and the number of the time and the emulsion stabilized assessed after the cycle. The deep phase separation, the rate of the degree of the phase separation is and the emulsion can be easily determined by the skipping the certain amount in the diluted cylinder and the measure the volume of the separated phase after the definite time the interval. The phase separation may be resulted from the creaming and the occlusions of the globules. The phase separation test can be accelerated by the centrifugal at the low and the moderate speed. This is the all the evaluations of the cream. The third preparation is the gels. Gels are the homogeneously clear semi-solid system consisting of the dispersion of the small or the large molecule is an aqueous liquid vehicle rendered the jelly-like by the addition of the gelling agent. Gels are the aqueous colloidal suspension of the hydrate that forms the insoluble medicament used for the medications and the lubrications. These are the gels preparation. The preparation of the gels, the gelling agents, among of the gelling agent used, the synthetic micromolecules such as the carbafor, carbamer 1934. Second one is cellulose derivatives such as the carboxyl methyl cellulose and hydroxyl propyl methyl cellulose. The natural gums such as the tagacan. These are the gelling agents that are using the preparation of the gel. The type of the gel phase, this is the figure of the single gel phase model and domain mosaic model. The single phase, the gel is the which is the micromolecules are the uniformly distributed throughout the liquid with no appearance the boundaries between the dispersed the micromolecules and the liquid usually involved in the organisms. Second one is the two phase domains when the gel's mass consisting the of the floccules of the small distinct particles usually involved in the organism. This is the flow chart of this the type of the gel phase. Second one, kind of the gels, the control release of the gels, bioadhesive gels, organogels, extended release gels, amphiphilic gels, hydrophilic gels, complexionization gels, the thermosensitizing solution gels, and the reversible hydrogels. There is the various kinds of the gels. The last slide we are the learn the type of the gels, the single phase and the two phase of the domains. This is the kinds of the gels we have to learn. Evaluation of the gel, spreadability. The modified apparatus consists of the two glass slide containing gel in between with the lower slide fixed to the wooden plate and the upper one attached to the balance by the hook was used to determine spreadability. Second one is the extractability. The simple method was adopted for the determination of the extractability in the term of the weighted the gram required to the extrude 0.5 cm ribbon of the gel into the 10 cm for the collapsible tube. This is the evaluations of the gels. This is the marketed formulation of the ointment or the semi-solid preparation. And last one is thank you.